Hi, my name is Jennifer Smith Parker, Director of Insights at Biospace, and you're listening to Denatured. In this episode, you'll hear from Mark Lowdell, CSO and co-founder of INmuneBio, and Vishwas Seshadri, CEO and Director of Abeona Therapeutics. We explore how recessive dystrophic epidermolysis bullosa has become a defining case study for cell and gene therapy and what this ultra-rare disease reveals about immune risk, manufacturing complexity, and the future scalability of advanced therapies. Hi, Vish, and hi, Mark. Thank you so much for joining this episode of Denatured. If you can please introduce yourselves, that would be great.
I'm Mark Lowdell. I'm the Chief Scientific Officer of INmuneBio, a card-carrying immunologist and cellular therapist since the 1980s, and the inventor of the drug we're going to talk about today, which is to treat the pediatric recessive dystrophic epidermolysis bullosa, or RDEB, as we prefer to call it.
Thank you, Mark. Hi, everyone. I'm Vishwas Seshadri. I'm the President and CEO and Board Director at Abeona Therapeutics. We're a cell and gene therapy company, and our FDA-approved product in the disease space of RDEB, which is patient's own skin with genetic modification to produce collagen 7. So very excited to talk about the disease as well as the solutions we're trying to develop for that.
Perfect. So that's a great introduction, gentlemen. So let's go ahead and start for our audience just to describe RDEB in the sense of what it is and what are its symptoms and how, basically, from my understanding, what a terrible disease it is.
Yes, so as Vish just said, it's due to a selective mutation or loss of collagen 7A, which is one of the critical proteins anchoring the dermis and epidermis at the dermal-epidermal junction. But it's a problem beyond the skin. It's a systemic problem. So the symptoms they have in the skin, which is sloughing, the slightest trauma causes the epidermis to slough from the dermis. They get blistering, but they get it up their nose, behind their eyes, down their esophagus. And so these children suffer tremendous pain and distress through the dressings that they need for skin. But they also then have these problems of failure to thrive because their gut doesn't work. And a lot of them get strictures because of the formation of fibrosis. And some of these are esophageal strictures. So they may end up having to have esophageal dilatation procedures every 2 or 3 weeks, some of the children that I've seen in London. So it is a very, very debilitating disease. And the median survival for RDEB patients, and RDEB being the worst of the conditions that form epidermolysis bullosa, but the median survival is mid-20s, and many of these patients die of squamous cell carcinoma. So some major problems that need skin treatments and some systemic treatments. But Vish knows more about this probably than I do.
Well, if I just add, maybe, I think you've summarized everything, Mark, about how debilitating the disease is. These large skin wounds sometimes have, especially in the extremities, they become chronic, which means they cannot self-heal. And once they become chronic, they have a high propensity for these squamous cell carcinomas that Mark mentioned. And oftentimes that can take the lives of patients, and sometimes even infections that get into the body through these wounds can also lead to sepsis and claim lives. And because of this, if you look at the cumulative risk of developing a squamous cell carcinoma for moderate and severe RDEB, by the age of 50, it's 95%. So that's how important it is to treat these wounds. And of course, it's a systemic disease because connective tissue is also beyond the skin. So the gut and even cornea are dependent on collagen function.
Okay. I mean, this definitely sets out a very, unfortunately, grim landscape for patients. But there's hope. That's what Abeona has, and also what you're developing, Mark. So we know that RDEB care has almost been entirely about wound care and symptom control. But how do you both see gene and cell therapy shifting the goal from managing symptoms to rewriting the biology of the disease itself?
As Vish said, it's a progressive disease. And so getting these children early and being able to treat these chronic conditions before they become life-changing is very, very important. And the patients that we've treated, the pediatric patients we've treated, we've definitely seen improvements, particularly in the children under the age of 10. So I think the future is looking at managing these children better with treatments. At the moment, we don't have a curative approach for collagen 7 deficiency, but giving these children the skin grafts that they need, giving them something other than just a topical cream that allows them to stabilize these wounds. When a study was done by the FDA and more recently in Europe, looking at over 800 children and their carers, the issues that came up as the two most important issues were itch and pain. And it's difficult to understand quite how pain is so important in the distress that these children suffer. But the itch drives this sort of itch wound cycle. And as Vish said, infection is a major problem. So trying to improve their neutrophil function to deal with these infections, trying to deal with the skin lesions, and trying to reduce the itch so that these children don't scratch, they don't make the wounds worse. I think essential to improving their livelihood.
And if I could just add, I think given the complexity of the disease and how much it affects in the body, one therapy or one approach is not going to solve all the problems. We're gonna need 3, 4, 5 different approaches acting in tandem to address these problems. But since we have been focused on the skin side, which is more of the external barrier, and as one of the focus areas, as Mark alluded, we're following these patients for both patient-reported endpoints like pain and itch, as well as investigator-observed level of wound healing and things like that. And in terms of how are we trying to even bend the course of disease that you asked, Jennifer, it's more of, can we give immediate relief, but also think of changing the disease in its grassroots? When I say that, I'm not saying that you're going to change the germline DNA makeup of these patients overnight, but can we at least give some kind of durable benefit with therapies that can supplement the gene in a way that their own cells can be enabled as factories to start producing the missing Col7 protein? And that's really the approach that we've taken, where we have recently presented data 12 years after a one-time therapy where there's wound healing and pain control that's been followed over the years. So that's encouraging. Hopefully that's one step, and then we get more and more therapies into the market to address this in a holistic way.
Yeah, that's actually such an interesting point. You're saying starting off over here with the skin grafts, but as you mentioned, Mark, that there's all these other aspects to consider too with pain. So just going back to the skin grafts, I'm just wondering about how do you see the challenges of manufacturing and delivering these grafts, both as engineered products and living immunobiological interfaces with the patient?
Yeah, I think the process begins with the patient's own cells, right? So we need a sample of their skin biopsy, and it's the topmost layer of their skin, which is epidermis, made of keratinocytes. So we extract keratinocytes, grow them in culture, and gene correct it, and then grow them further to make these sheets. And that's really what we supplied back to the centers, where a plastic surgeon would apply these sheets like any skin graft on the patient, and that becomes part of the patient's skin over time. So that's really the therapy. The process takes about 20, 25 days to manufacture and supply these grafts to the patient. And a lot of because patients have to travel to these treatment centers, it's not going to be in their home clinic or very close by. But we as a company are supporting those types of logistics just to make that process a tad easier for the patients as much as we can. And the clinical data have been quite impressive and promising. It's a ray of hope for these patients that if you can achieve long-term wound healing and pain reduction for some of these menacing wounds, then that's seen as worthwhile going through this process or logistics of having to go to a center, give their skin sample, and then wait for an operating room date and basically configure their lives around that process for that period window that they have to plan around, right? So that's how we are hearing from the patients, and they have a very strong network. These patients are very well connected. They have their Facebook page. So one patient talks about their experience, and that's really how the community depends on those experiences to learn and come forth for these types of therapies.
The patient support groups are really central to this, and it's exactly what Vish described. The patients hear about treatments or clinical trials, and it runs through the network very, very, very quickly. So yeah, it's a patient group that's easy to work with in terms of access, but difficult to work with because of the clinical condition that they've got.
Okay, understood. Are most of the patient populations, is it just inevitably going to be in the U.S. or, and Mark, where you're based out here, how is it in the U.K.?
So the prevalence of RDEB is pretty much flat across the world. And so we have upwards about 100 RDEB children under the age of 16 in the UK. And we're fortunate from my perspective in that we have 2 national centres that are funded by the healthcare system here. So nearly all of them go to the 2 national centres, which makes it relatively easy to treat them and bring them into trials and bring them into early treatment. But yes, if you look at the population size, that's 100 children in a population of 60 million, and if you just multiply it up by the US population, you get the US numbers and the European numbers as well. So I was speaking to RDEB in Germany, the Deborah, the charity in Germany, and they came back with very similar prevalence. So yes, it's a global disease, and talking to people recently and trying to get drug into Russia, into the Middle East, it's pretty much the prevalence is the same globally.
Yeah, that is quite interesting. Now, I'm just curious here when it comes to this entire cell therapy platform that we're looking at right now. So a decade ago, cell and gene therapy, maybe probably more than I said, even a decade ago, was still very novel, especially for an ultra rare disease. But I'm wondering, how has a real world experiences in RDEB, both clinically and immunologically, changed the way you talk about risk, safety, and benefit with regulators, investors, or families?
Well, Vish has had more experience than that, so I'll defer to Vish in the first instance. But
yeah, I think when we talk about real world experience, right, I think, of course, since the launch of our product, it's very early days for us as well, because we have not yet commercially followed patients for years yet. But I would say that when you brought up this word risk, how is risk viewed by various stakeholders? I think let's start with the patients, which are the most important stakeholders. I think when we say, what is the risk of treating patients with a cell therapy or a gene therapy or both, we always have to ask, what is the risk of not doing that? That is much bigger than the risk of trying something new. It's always going to be the unknown, the uncertain. Will this technology work? Will it introduce a new risk? But then if you're reasonably satisfied that those risks are minimal compared to if you leave this untreated, patients are going to have a very poor quality of life and will also have very bad life expectancy. And I think that is the context that at least the regulators have viewed this as well, because our technology is introducing a gene and it's autologous. I think when we use autologous cells, it means the patient's own cells that can minimize the immunological rejection factor, so you don't have a graft versus host kind of a problem there. But we are still using vectors that introduce these genes, and they are highly regulated. We have very regulated ways of seeing, are we doing something unintended, and how have we taken care of that? Deborah is one of the organizations that's patient-focused, there's a patient community there. They say the cost of doing nothing is humongous, right? That's really what the regulators also hear. And when we talk to the FDA, we also take these representatives from the patient communities with us to explain that look at risk in relative terms of doing versus not doing. So that's one. I think you brought up the investors as well. I think there has been a lot of investor participation. If you look at RDEB, the therapies, right from the topical gel that was approved, and then there is a bark extract that also Filsavez is one such product. There are so many products that have come to market, and that's testimony to how investors look at this unmet need and where there's an opportunity. So I think there is a positive change, I would say, overall, looking at how do we assess risk, both from a financial perspective, but most importantly, from a patient care perspective, right? So that's my take. But, Mark, please chime in. You have a different angle to view these.
I think you've hit the nail on the head, actually. You're talking about risk, and sort of the gene therapy risk that everyone got worried about were the children treated in France in the late 1990s for severe combined immunodeficiency syndrome, another disease that is dreadful and kills children. Now, that was cured by a gene therapy, an autologous gene therapy, and two children developed acute myeloid leukemia because the virus, the lentivirus or the retrovirus, chemoretrovirus, integrated into the wrong site in the gene. Now, I knew the clinician treating those children. It shut down gene therapy for years, but in reality, what it gave, you took children with a fatal disease and you gave them a disease that could be treated, because acute myeloid leukemia in children is actually a very treatable disease. And both of those children were treated and were cured of the acute myeloid leukemia. But it put back gene therapy for a long time and created this fear, oh, what about inappropriate integration next to a driver, whatever. And that, I think, for our discussion with regulators, obviously, this particular therapy of mine is not gene modified, but I'm a professor at University College London and involved in all sorts of developments. We have discussion with regulators all the time, and they reflect exactly what Vish has said. They're much more comfortable with the risk that we take depending upon the treatment you're trying to deliver to the condition that you're doing. And it has to be a risk-based approach where actually you can take a bit more risk. You wouldn't be doing a gene therapy for dermatitis, for example, but a gene therapy in this scenario is entirely appropriate, particularly given the safety that we've developed or understood in the last 25 years.
Yeah, I think it makes total sense what you're saying. You really have to look at the risk-benefit profile here.
We had a meeting with our UK regulator a couple of weeks ago, and one of the comments we got from one of the parents of the children who were in the last trial that we ran, so we're asked, you brought this child all the way, several hundred miles to come in to be treated in London. And the parents said, if I can improve the life of my child by 1%, that makes actually a meaningful difference. And you think about when you're developing drugs of 10%, 20% improvement, so 1% will get my child out of bed. And that's why we will do it. And that's why we'll take this risk. And I think that speaks to it very well. We don't need to see huge change in these children to have a big clinical change. And that's one of the problems one has when assessing the trials. You know, how far what do you really need to show to have a meaningful clinical benefit?
Yeah, that's incredibly important. That's something I remember to covered in the years past when I was a journalist, is that sometimes a disconnect between clinical significance and statistical significance, or between patients want to see and what regulators want to see can be different. So I think just to sum it up here, gentlemen, if you had to pick one or two lessons from RDEB, let's say coming from the cell and gene therapy perspective and clinical perspective, and one from the immunobiology and the translational perspective, what would you say is the most important takeaway for how this avenue of therapy will expand into broader patient populations over, let's say, the next 5 to 10 years?
I would say some experience with this product has definitely taught us a lot, right? These are complex biologics. I think engineered cell therapy in itself is a new field relatively. And within that, to have something like engineered skin cells that are sheets of skin cells, it's even more sophisticated. But I think the experience of taking such innovations from academic institutions like Stanford, after having done a clinical study and where manufacturing was done in those institutions, and then we bring it up into industry and we try to scale that, I think the most important thing is staying close to patients and their needs and the community, because how we build a product and the characteristics that we care for in the product has to be driven directly by what their lives looks like, and are we really solving their problems, right? That interconnectedness is important. I've never in my career had an experience of having met, after the trial is done, 60% of our trial participants and had dinner with them over some events like these. It's not something that you will see in large indications like when you're working on lung cancer or something like that. It's a very special thing. And I think the best we can take from that is learning how can we best serve these communities, what are their needs, and building that into our product. I think whether it's cell and gene therapy or any other modality, right? That's, for me, the biggest learning. And it's a continuing journey. I think once you go scale up and now go into the market, you're going to only have more and more of that because you're implementing it in a commercial setting. That's a completely different ballgame. So, yeah, that's where I would leave my thoughts. Mark, we're happy to hear yours.
Yes, I worked in hospitals and academic units all of my career, and it was have been tissue engineering, immunotherapy products. And the take-home message I have is one, as Vish said in their setting, you have to have a drug that can be delivered in the environment in which you're delivering it. Now, they have specialist treatment centers, which works extremely well for a complex product. We have engineered our product to be very friendly to hospital pharmacists, so that it doesn't come in as a drug that's any different to any other drug, because hospital pharmacists get very nervous about cell and gene therapies. So that's number one. Two, we've made it as friendly to the end user, to the clinician, so the clinician wants to use it. Dermatologists don't do cell and gene therapy, so you've got to get them on board and get them to understand that this is not crazy. So you make it in a way that can be delivered. Vish has done it in a way that can be delivered by plastic surgeons, and they get a product that they're familiar with. So we deliver our product in a way that the clinicians are used to it. Ours comes cryopreserved, so it comes with an automated cooling device, and you press the button and it goes ping and tells you, yes, now's the time to give it to the patient. And then we've worked with the patients to say, these are the technical difficulties. You're going to have an intravenous infusion, given all the problems that they have with venous access. But we've worked on needle sizes so that there's less problem with that. So it's about, making it fit in the real world with the environment in which you're going to work, So it's a great community. And as Vish said, meeting these families, you do become really involved with them, and they want to come back and they want to be treated. So I think, yes, it's about working out how to make it work in the real world for the patients and the clinicians and the hospital environment you want to use it.
Well, that has been a great introduction for audience who wasn't aware for RDEB. And I love that we're ending on a very positive note going forward here. So there's a lot to look forward to for the future and for patients. So thank you so much, Vish. Thank you so much, Mark. If you'd like to listen to more episodes of Denatured, please turn to biospace.com. Thank you.